and CDAST University of Jember, and today we continue to the invited speaker lecture will be conducted by Mr. Dr. Didi Pujarestanto, and we invite Mr. Didi Pujarestanto. Come to the stage. Time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, today the last presenter invited lecturer. This have the four presenter. Maybe I want to call one by one to bring the presentation, and we have just 10 minutes to presentation, and the first one I call Dr. Siti Nurbaya Uslan. Time for yours.
Okay. Yeah. We start with the song. Hi. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Hopefully, you have a really good and enjoy day today because we have been walking for quite long distance this morning. Okay. So for today, I'm going to share with you our research about yeast expression system. It's quite different than other people who are working with the proteins and everything. Now I'm working with the host. It's a host where you can use to express your protein of interest, any proteins, as long as it starts with DNA, a gene, then you can include into this expression system. So I'm working with a yeast named Microbial strain SO. Be specific to strain SO. Strain SO stands for Sprout Orange, not Siti Oslan. Even though my name is Siti Nurbaya Oslan. Okay, so the title will be a quick and reliable Microbial as an alternative host for recombinant lipase expression. So this host is used to express lipase, but it's possible to express other recombinant proteins. Okay. Next. Tekan bawah. Okay. So what we have today, normally when we want to express our protein of interest, you will select prokaryote or eukaryote expression system. So prokaryote normally you will select E. coli, Lactococcus, Lactobacillus, Pseudomonas, and then for eukaryote we have yeast named as Saccharomyces cerevisiae, Pichia pastoris, Hansenula polymorpha, and all this. And then for plant we have tobacco, Arabidopsis, soybean, and insect cells, Drosophila, and mammalian cells. These are common expression hosts available. Okay, so and then for this study we are interested in yeast. And then why we choose yeast? There are a lot of candidates over there. Then why we choose yeast? Because yeast have strong promoter. It can grow to high densities, much higher than E. coli can grow. I'm not ban any E. coli, but this is the strength of yeast. Okay, and then it is required inexpensive substrate to grow. And then it produces glycosylated heterologous protein, where prokaryote cannot do this post-translational modification. And then it's easy to scale up in fermenter, and then do not require antibiotic for protein production. This is because once you have designed your vector to be integrated into the genome, that means you don't have to supply the antibiotic. It is all stable. And then after that, yeast can provide creative and reliable approach to express difficult protein. For example, if you have protein from mammalian cell or higher eukaryote, this is like a platform. You can choose either you go to E. coli or eukaryote, but this is like in the beginning. If you go to higher eukaryote, maybe you can have more difficulties and everything, but this one is just starting of the eukaryote. And then yeast strength is able to secrete protein into culture medium. If you use E. coli here or other Yeah, especially E. coli, you need to use bacterial release protein to release the protein out. But for this one, we have the secretory signal so that the protein can be secreted into the culture medium. So that your target protein will be in the culture medium, not in the cell. But definitely yeast can also express your protein of interest in the cell, but it also can secrete into the culture medium. Okay, so like I told you earlier, it is first isolated from spoiled orange, okay, actually from night market, from Serdang, okay, close to UPM. This is a famous night market close to UPM. You're most welcome to go to UPM and join our night market. Lots of food. Okay, so other than food, we offer yeast. Initially, it was identified as Pichia species. And then it closely related to Pichia gourmandi with 99% similarity if we use internal transcript species identification, molecular identification, and then the sequence has been submitted to gene bank. And then, okay, so for the promoters, if you want to choose, if you want to clone your gene of interest, you need a promoter, right? So the promoters available in this host is alcohol oxidase promoter and formaldehyde dehydrogenase promoter. So for E. coli, normally you use T7 promoter. So this one we use, this promoter represent the metallotrophic promoter. Okay, so this yeast has been used as expression host. It's able to express thermostable T1 lipase. For Roger, like other people yesterday keep talking about T1 lipase. So I'm using T1 lipase as a reporter gene, a reporter protein to test this expression system because we do not have problem with T1 lipase. So we want to test that this bacterial gene can be expressed in yeast or not. So now, initially this T1 lipase gene has been expressed in E. coli, followed by commercial system, which is Pichia pastoris, and then after that it is transferred into local host, which is strain SO. Okay, we use PPZ alpha B, which contain alcohol oxidase promoter. And then this is the optimal condition. I'm going to explain about this later to compare this system with Pichia pastoris expression system. So this is the overall pathway for metallotrophic yeast. If you claim your strain is metallotrophic yeast means the yeast is able to use or consume methanol, use methanol as the sole carbon source to start this pathway. Okay. So to further identify this strain SO, we use assimilation test. And then it is from this result, all these carbon sources tested, it is confirmed that this species is Merozyma gourmandi. Okay, it's similar to Pichia gourmandi, but it is now some people name it as Merozyma gourmandi. Okay. So this yeast has been filed for patent. The host, I mean not the yeast, I mean the whole system, the method to transform and to clone the gene into the system, we have filed. And then we start with amplification of T1 lipase, and then we clone into this vector, PPZ alpha B, and then we use electroporation method to transform the recombinant vector, and then last but not least, we express into this broth. Okay. Okay, if we compare here, strain SO versus Pichia pastoris, which is the commercial system just now. So in terms of the medium, they use similar medium, YPTM medium, yeast, spectrum, tryptip, soyberos, and methanol medium. And then they with different initial mass. For strain SO, it requires higher mass, higher initial mass, which is OD22. Then the rest require only 7. Methanol induction is lesser, much lesser than Pichia pastoris, which is 2%. This one only require 1.5%. For inocular age, this one is similar to the previous one, to the initial mass. Optimum time, if you look at here, we have 30 hours here of production time. If you compare with the Pichia pastoris, with different strain of Pichia pastoris, this one is like 192, 144, 144 hours. So you need to remember that methanol here, even though it's just 2% and 1.5%, it plays a really significant role where this methanol 2% need to be supplied to this host every 24-hour interval. So if you have 30 hours, that means you need to supply only twice, right? At 0 hour plus 24 hour. But this one you need to 0, 24, 48. So you see a lot of methanol required when you want to produce in Pica pastoris. Okay, so if we, but now we have problem with the production with the lipase activity. It is much lower. But if we convert to unit per mil per hour, within 1 hour you can get this one is much better than, is higher than the Pica pastoris. Okay. Okay, so you can see the difference between growth rate 22 and 7 just now, right? Is there any difference in terms of growth for these two systems? No, it is same. So Pica pastoris, we have it reached its stationary phase around 18 hours or something, but same goes to Merozyma graminea. So that means this in terms of growth, they are just the same, except the way they behave to express the recombinant protein, they are different. Even though we have used similar vector, similar induces, similar genes. Okay. So okay, what I want to highlight here, if you want to have 1 gram of protein, this system offers 3.3 times faster than Pica pastoris, and then with 4.4 times lower methanol, it saves you a lot of cost, okay? And then after that, okay, the uniqueness about this system, something new that we found, this strain does not require methanol. That means even though you do not supply the methanol, you can still get the recombinant protein expression. This features is absent in the commercial system. So in commercial system, which is Pica pastoris, no matter what happens, you need to put methanol. Otherwise, there's no expression at all. But this one still you can get this one. You see, straight SO without the lipase here, and then if you have straight SO with the lipase, you can see the fluorescent color here. It shows that this lipase is present in this system, in the expression. It's happening. Okay. But you know what is? I can speak faster. Okay. So in order to understand what's going on, we check on the metabolite. Maybe there is some byproduct present in the host that can contribute to the expression. So we run for the GCMS, which is untargeted metabolite, because we don't know this intracellular metabolite. We test at 0 hour versus 60 hour. So you can see here different products present after 60 hours of cultivation. Why 60 hour? Because 60 hour, when we repeat the experiment, 60 hour give you more expression. The 30 hour just now is with methanol. Now we are talking without methanol. If without methanol, you need longer time. Okay. So we also tested for the extracellular metabolite. Nothing much significant here. Okay. So because everything coming from the inside, outside is coming from the inside actually. So outside is just the medium. Okay. Okay, so when we check for the draft genome, Enzyme draft genome, it is reported by ATCC 6260. The features here, you can see here that the genome size is about 10.61 with a protein, almost 6,000 protein. Okay, and this is different than Pica pastoris. Okay, so we want to understand why it is different than Pica pastoris. Then we check on the alcohol oxidase protein, because the alcohol oxidase protein is a key enzyme. When you put the methanol, this protein will catalyze the methanol, metabolize the methanol. So what happened here? We used hidden Markov model by using alcohol oxidase protein to build the model, and then we search against the reference genome. And then what we found, instead of short chain, methanol is short chain fatty alcohol, and then we found it is a long chain of fatty alcohol, not a short chain alcohol. So the difference between long chain alcohol compared to the alcohol oxidase present in Pica pastoris is only 14.8%. That means it is different. Long chain is different than the long chain methanol. Okay. So when we build the phylogenetic tree here, short chain alcohol oxidase from methanotrophic yeast, they are all grouped here, but long chain, they are separated. Okay. So where we are up now? So we have used the host to express other proteins such as amylase, diamino oxidase, and protease using alcohol oxidase and formaldehyde dehydrogenase promoter. So both of these promoter do not require methanol for induction, actually. Okay, so we are also done to check all the metabolite that override the alcohol oxidase promoter, and then we are doing some little bit of comparative genomic study between strain SO versus other metotrophic yeast. We want to see in detail at the genome level, and then we want to develop a new engineered protease deficient strain SO. Maybe the protease actually prevent the higher expression because it might degrade. Because this is the wild type. Normally Picapastoris, they have engineered the strain to express your protein of interest much better. Okay, as a conclusion, wait. Okay, so megazomal remedy can be used to express using alcohol oxidase promoter, and these two promoter, it is a novel yeast expression system without methanol, and then it can produce methanol-free recombinant protein if you do not induce with the methanol. And then whole genome sequencing and metabolic analysis can be pursued to get the full information about this system, because we do not have any reference to use this system as expression host. I'm sorry if I'm talking about like a bullet train, but it's about to finish now. Sorry. Okay. Next. Okay, this is our picture last 2005, having happy bowling. Maybe this time I won best female bowler, I think. And then I would like to thank my student, my Probaka. He's not here. He's my supervisor during my PhD student, and then Raja, also my co-supervisor. And then I want to thank Dr. Adam, Dr. Suriana, Arpa Abu from Universiti of Malaya, and then Dr. Ain from UKM. These people are doing my job actually. Nida, Apple, Wani, Mary from Nigeria, and Azila. So I would like to thank my research grant and sponsors. I think that's all. Thank you.
Thank you very much. Please stay in this front. The second speaker is Dr. Trihandoyo. Maybe maximum 15 minutes.
Just 10 minutes.
10 minutes. Okay, thank you. Time for yours.
Thank you very much for Pak Didi, yang terhormati Bu Raja dan Pak Bambang. I would like to share my research, maybe very different with all of you. I would like to explore my Indonesian biodiversity of Indonesian aromatic and black rice. Why? Next, please. Di-off-kan bisa. This our backgrounds. We have many kind of rice in Indonesia. In this case, I focus just for aromatic and black rice. The problem of Indonesia, actually we have increased the people many years, high fertility, and also issue problem. In this research, we try to analyze the morphology, physiology, and also molecular and also physicochemical of rice for new premium rice and also standardization of rice. Why we need the standardization? Sometimes issue we eat like rice plastic, do you know? Rice plastic and something synthetic rice in Indonesia, and every day we eat plastic, something like that. That's why we need the standardization of rice. Okay, next. This is our map, and many kind of aromatic rice developed in my country. syllabus, Sumatra and also in Java. This most rice developer here. Next. The first research did by my student is we check all the aromatic rice in Indonesia. Morphology and collecting, and next, and we try to RIPD to make some groups by similarity matrix, and next, we got this picture. After RIPD analysis, we got the 3 group of rice in Indonesia, and different, maybe different taste, different morphology of plant, also grain. There is explained in the next slide. After we collect all the aromatic rice in the different groups, we try to make a new premium rice. Actually, for the first time, I tried to black rice, and we develop culture to get a haploid and diploid. Next, this mature anther. Next, this picture had a mature anther. First, we optimize, because seems like easy to do anther culture. This is for the first time for me, and make my student more stressed here. This is a problem. After my student stress, and next me. We got the colors induction until now. We tried to 5 cultivars, and we got nice colors. Okay, this from the 5 varieties. Hitam Malik, yeah. Hitam means black. Malik ini sangat hitam gitu ya. Pari ireng, actually this is the same name, but different location. But I'm not sure this a similar genetic or not, because we already separate in 4 groups. This is a process, different process between each varieties. Okay, next. After we got the colors, actually I'm not success to regenerate to plantlet, but we found like this potential colors for anthocyanin production. And maybe in my dreams next, we can produce the anthocyanin in the colors levels. And I hope this extracellular metabolite secondary, and just extract the media and maybe separate by metric, like gel filtration or ionic metric, and spirit produce mass anthocyanin production. For standardization, this is more important information. For example, this ongoing research. First, last week ago, I tried to use scanning electron microscope to rice grain, and I got this like picture. This just one. Saya ambil dengan paksa ini. Jadi, this picture maybe a bit between rice, different rice, maybe different structure, three-dimensional structure. And also, we try also a gelatinization process to make a standard for premium rice. This most important because the amylose and gelatinization process is a key of the quality of food. This is the last for me. Because when I read your book yesterday, just what I say, men maybe I can just 5 or men, but when I open all of the book, all women. Di balik pria yang sukses terdapat wanita yang hebat. Next, I greet thanks to my student. Yes. He always give me support to do research. I hope they come here. Almost of my students is a woman. And this research supported by Ministry of Research Technology and Higher Education, and they support me and get 5 grant in these years. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Okay. The third speaker, Dr. Fairul Niza. Please, time for yours.
Assalamualaikum and good morning. For today, actually, especially for this colloquium, I decided not to present something related, not much on enzyme or either microbial, but I choose something that I just started on, which is related to plant, which is the palm oil. So palm oil is very close to both Malaysia and Indonesia. So for today, especially for this colloquium, I'm going to present on a preliminary studies on this defensin from palm oil. Okay, sorry. All right, so a little bit on the plant defensins. So defensins are actually a cysteine-rich protein. So the size is around 45 to 54 amino acids. So this is one of the largest families of antimicrobial peptides from plants. And it is part of the integral part of innate immune system for plant. And it is found as an ancient system in all multicellular organisms. So not just plant, but all of us have defensin as part of our innate immune system. But for this project, I'm going to focus more on the plant defensins. And for plant defensins, there are many reports showing that they produce antifungal activity, antibacterial activity, proteinase inhibitor, and also insect amylase inhibitor activities. And these plant defensins are also called as promiscuous proteins because they can do so many activities All right, so this is just a photo of one crystal structure of plant defensin that has been reported. All right, so a little bit on the mode of action for plant defensins. So the famous one is they form pores on the microbes membrane, and also they can also inhibit the microbes protein synthesis and the enzyme activities. So it means that they are actually enzyme inhibitors, especially for amylase and also proteases. And also these defensin can also deactivate toxin by protein folding. And these are some common characteristics of these plant defensins. So yesterday, someone mentioned about mazhab, NMR, mazhab crystallography, and mazhab modeling, right? Computational structure. So for today, I'm going to talk about these plant defensins has been solved by all three methods. So it doesn't matter which mazhab that you are holding. So for this purpose, I'm going to be very greedy. I'm going to grab all the mazhab to get what I want. Okay? So because of the size of this protein, because sometimes they say it's a little bit too small to be a protein and a little bit too big for a peptide. So in order to solve the structure, the researchers are using any ways that you can. So you can try out crystallography, you can try out NMR as well. So for this project, I start the work by doing computational modeling. All right. So the common structure, so it is a well-conserved 3D structure composed of cysteine stabilized alpha beta motif. So it has one of alpha helix followed by 3 anti-parallel beta sheets. And they have 3 to 4 disulfide bridges, and some reports show that they can also come in with 5 disulfide bridges. So imagine how stable this structure is, with very small but very stable. All right, so these are some of the reported structures of plant defensin. Few of them has been elucidated by X-ray crystallography, and also some of them has been elucidated by NMR. And back to the topic, because I'm interested in the defensin from the palm oil plant. So this is just an overlook on the overall oil palm estates in Malaysia. So we can see that almost everywhere in Malaysia we have the estates of palm oil. And these are some facts or figures on the palm oil production in Malaysia. So we produce 39% of palm oil production in the world, and 44% export in the world. So there are reports on this defensin. This oil palm defensin has this biological role in oil palm defense in response to a pathogenic fungus, Ganoderma boninense. And these Ganoderma boninense can cause a severe disease for these oil palms, which it can cause the basal stem rot. So these are the fungus, the Ganoderma, and these can cause the palm oil tree to the stem area to be rotten, and then later the tree will die. And this will cause around 46% reduction in oil yield. So it's a big issue in palm oil plant industry. So back to the defensin. So this defensin, it has 231 nucleotides, so with 71 amino acids. So a little bit too big for a peptide. and it has a potential signopeptide. And they have 8 highly conserved cysteine sites, usually found in plant defensin. And for the mature part, it is lacking the 30 amino acids residues. So this mature part has been cloned into E. coli system, and it showed as expressed as a soluble recombinant protein, and it was proven that it is a highly thermostable peptide. And this defensin has demonstrated inhibitory activity against the growth of Ganoderma boninens by inhibiting the starch assimilation. And from this paper, it proposed a model which says that this defensin inhibits either the amylases produced by the palm oil, or it can be also the inhibitor of the calcium ion channel of this Ganoderma boninens. All right, so the problem statement for this research is, as I mentioned just now, this defensin is a highly stable peptide, and it has a high potential to be developed as a novel antifungal drug because of this inhibitory activity against Ganoderma boninens, the pathogen. However, this fungal activity of this defensin has not been elucidated, and we don't know how exactly the activity mechanistically or structurally. So this is what triggers us to do some research on it. So these are some of the research questions. So we want to know, how does this defensin inhibit the alpha amylase activity? And also, and now you can see it has some relationship to enzyme, to EMTech, right? Slowly I'm going back to EMTech, not running away from Prof. Raja, although I'm doing plan now. All right, and also another question is, what are the critical amino acid residues involved in the interactions between this defensin and the amylase, as well as during the inhibitory activity? So we would like to know on this specific subject. So we came up with this hypothesis. So we think that by predicting the 3D structure of this defensin, we can actually study the interactions between the defensin and the alpha amylase, and also we can solve the mechanism of the inhibitory activity of this defensin. So we come up with 2 small objectives just for the preliminary research. So the first one is to predict the structure using homology modeling, and the second one is to search for the possible docking pockets for the ligand binding, the ligand protein interaction, and later we do the docking as well as some molecular dynamics work. Next, so this is the simplified version of the methods. As I mentioned, we check on the sequence, and then we find the template, and we predict the structure, and then we run some molecular dynamic simulation, and we do the docking. So these are the results on the multiple sequence alignment. So from here we can say that we can find the conserved cysteines in this defensin the same with the rest of other deposited defensins. So, and also we can see that it has the same secondary structure, almost the same. Next. And this is what we came out from the homology modeling method. So we can see that the structure in the ribbon as well as the atom residues. So we can see the disulfide bond in yellow color. If you If you look carefully, you can see there. So you can imagine that there are 4 disulfide bonds. Next. Okay, so enough about the structure. And okay, so for the MD, so we did some simulation on the MD. So we can conclude that this defensin is very thermostable. We managed to run the simulation up to 150 degrees, and we can see that the fluctuations were not that huge. So they can remain stable until 150 degrees. And also the RMSF shows that only certain parts of residues are fluctuating, but they are all the same when you compare with different temperature up to 150. So, and we did docking studies just to check on the binding pockets and what are the amino acids that can bind to this defensin. We tested them using 2 alpha amylases. So one is from Aspergillus, a fungus, and another one is from bacteria. And then we found out that there are 11 and 14 clusters of possible binding modes for the Aspergillus as well as Bacillus licheniformis, sorry. And this is just on the surface, we can see the docking, the interaction between the amylases and the defensin. We run this using HEDOC. And these are the amino acids that interacting with the defensin. And this is from amylase from Bacillus. And some conclusion. So the predicted model showed high satisfactory evaluation scores, so it is accepted. And the MD simulation also proved that this defensin is high, it is very thermostable. And when we did the docking, we can it shows that this there are interactions between the defensin and the alpha amylases, and they form hydrogen bonds with this catalytic residues. And also we can conclude that this study provide preliminary data on the interaction of this alpha amylase defensin complex, and it can be useful for the production of the defensin as antimicrobial agent. So future work, we will proceed this work with some characterization on the inhibitory potentials to determine the inhibition type. Sorry, there's a typo there. And also we would like to determine the binding constant of these protein ligand interactions by titration activities or experiments. And also we would like to elucidate the actual 3D structure. So if it can be solved by X-ray, all right, or if it cannot, we can try and use NMR because the size can be elucidated by both. So something that the future for this defensin. So we are thinking of maybe cutting them into smaller sizes of proteins and checking on which are the sites that can function as antifungal, and also it can be implemented as in transgenic plant technology, whereby you can enhance the defense mechanism for certain plants, or maybe for the palm oil plant itself. So you can double up or triple the defense mechanism by introducing the transgenic plant with this gene. And also maybe we can come up with a simple alpha amylase inhibitors, because from these from the previous activities or the previous report, they showed that this can act, this defensin is acting as an inhibitor, alpha amylase inhibitor. And a bigger picture again for overlook. So maybe this plant defensin should be not just focused on palm oil. It can be contributing for the development of transgenic plants expressing antifungal defensins, or maybe the production of defensin-based fungicide. So, but then again, it depends on all these planning or research direction. It depends on the determination of the regional, whether it is a priority for the country or for the specific fungal targets, so that this type of product can reach the markets. Because up till now, there are still, although there are many work on plant defensin that can show antifungal activities, but none of these defensin has been commercialized into a real antifungal drug. So yeah, so some acknowledgement, Prof. Raja, and my collaborator, Dr. Ho Chai Ling, for our plant people, and my temporary research assistant, Dr. Nurul Aini. Thank you.
The last speaker is Dr. Paravita Dewanti. Time for yours.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody, especially from Prof. Raja and Prof. Bambang. Today I want to share about my research. This is my topic, free virus synthetic seed production in sugarcane, growth and plant regeneration from embryo cell. This research is part of Professor Bambang research. So I think this is in a research group, Prof. Bambang research group, sugar group, and this is I want active in CDAST and agriculture. Okay, this is my introduction. In sugarcane, there are one problem about sugarcane mosaic virus, because the sugarcane mosaic virus can attack the in plantation. This is a symptom in leaf of sugarcane. There are a strip with a yellow strip in the yellow strip in leaf. So the effect is, if the sugarcane mosaic virus can attack the sugarcane, the production sugarcane decrease around 30 until 50%, and may even reach 80%. So, how to produce healthy seedling organizing for the disease-free seed stock? We make elimination SCMV by chemotherapy, especially in antiviral or antivirus, and until get free virus plantlet. And then we can develop two somatic embryogenesis for multiplication, and the last target is synthetic seed. Okay. The production of health seedling a large quantity can be done through somatic embryogenesis technique. There are many advantages for we get somatic embryogenesis, like this, maybe quick and easy scale up liquid culture, and then long-term storage, reactive preservation, production of somatic seed by encapsulation of mature somatic embryo, and the other. Many factor effect of occurrence of somatic embryogenesis and artificial seed are growth regulator, a source of explant, nitrogen, carbohydrate composition, or sugarcane, and sodium alginate. What is the artificial seed? Artificial seed is artificial encapsulation of somatic embryo and any tissue which has ability to form a plant in vitro or in vivo condition. Maybe you can look at this picture like this. This is a somatic embryogenesis and encapsulated by alginate. The purpose, there are advantages for the artificial seed, resistant to breakage and can be stored for long period without loss of viability. The purpose of the study are provide synthetic seed sugarcane in large quantity, which can be stored to suitable substitute the supply of conventional sugarcane seed, and then produce large amount of uniform and high quality sugarcane seed free or viral disease through somatic embryogenesis, and the last produce technology for making superior sugarcane seed free of virus. This is my roadmap. Maybe the long year I can get my target. My target is mass production synthetic seed. This is the last product in this year. The first time we work about elimination virus and callus induction, and then proliferation and regeneration, and the next liquid culture, multiplication, and encapsulation, and the last acclimatization. This is my method. This is a long method maybe, but this is a plant material. The material we can get from spindle leaf or leaf roll from plantation, and then we induction, callus induction. This is called explant, and then we growing callus induction proliferation until we get coleoptile. After we get coleoptile, we can encapsulation, germination, regeneration until we get the plantlet. Plantlet is a plant with complete part, root, leaf, and stem. Okay, this is a composition of media. There are many media in here for indoxycallus, different with proliferation, encapsulation, and regeneration, because depend on the stage of development. This is a result, percentage of virus elimination at different acylcophil and ribavirin concentration based on ELISA. This is we can cut off value if we get absorbance in the lower, so lower than cut off value, so we interpretation is negative. So percent eliminate until 10%. And different is in the upper cut off value, so we can the interpretation we get positive. It means we in cell in plant there are many virus in there. This is a confirmation by RT-PCR. We can get this is no virus, but but in here it detected for virus. And then we can enter to induction callus. There are percentage for induction callus. There are two variety in here, SUT02 and PSXX1, and time for callus. There are different media. You can look at this A, MS, Murasi and shock, Murasi and shock, basal medium, and then there are at 240 and like that different concentration. This is result for time of shoot induction, plan number until plan 8. Okay, this is a morphology callus in different composition. This is A, B, C, D, E. It's different medium. Now, this is a development for callus because for in somatic embryogenic we can we start for globular phase and then scutellar, coleoptillar, and then cotyledon, early cotyledon, late cotyledon until we get shoot. And then this is maybe it's same with this is analysis histological observation about sugarcane. There are somatic there are non-embryogenic and embryogenic. Maybe different because we can look at the non-embryogenic, maybe the large size in cell, but in embryogenic, this is a small size in cell. In this picture, we can look at about the morphology embryogenic and non-embryogenic. There is a different character about embryogenic and non-embryogenic. Callus and embryogenic, there are have a character variable and then easy to lose, then glossy, maybe different with non-embryogenic. So we can my target is embryogenic cell. We can look at this embryogenic callus induction at 6 weeks. There are in 6 weeks we get until 92%. Okay, in the next picture we can get develop of somatic embryo states in morphology and histology. This is a different states. This is pro-embryo mass structure, and then globular structure, and this is a scutellar structure. And the right picture, you can look at this liquid culture in different media. You can look, this is browning, this is a good liquid culture. And then after, okay, and then we can growing in the liquid and in the solid media until we get plantlet. So this is, I think it's same, but different treatment. And then this is the research about scutellar formation, coleoptillar, number of coleoptillar until percentage of plantlet. This is this is for the same as BAST. In the last, we can encapsulate it until we get synthetic seed, and then we can growing at or germinate in the next or in the different media. And below, synthetic seed percentage of germination for 4 weeks after storage period. This is my treatment, PEG, with different concentration. Okay, this is in liquid culture too, but different research. And this is a histology suspension. This is from reference. This is my result. Yeah, okay. Okay, this is last PPT maybe, last slide. This is a synthetic seed with different and alginate concentration, natrium alginate concentration. This is day of germinate, and then what's number of percentage, and this is and on 58 days. This is my conclusion for callus induction, proliferation, and regeneration. I think I have been get best medium. The best medium for alginate concentration for encapsulation, 3 until 4% sodium alginate and 100 millimolar calcium chloride. And the last virus-free synthetic seed and sugarcane have been successful for develop. Their advantages include being obtained the large quantity, take a relative short time, can be stored and facilitate to distribution. Thanks for Prof. Bambang and my student to help my research. Thank you for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Nice presentation. No. We still have maybe in the 7 minutes for discussion. Maybe any question? Please. 1, 2, 3, 4, 5. Enough. Please, number 1.
Thank you, moderator. My question to Dr. Tri Handoyo. As we know that in Indonesia there's many kind of the genetic biodiversity. To make a new variety, there's need a long time with conventional breeding, but with biotechnology, maybe it got time, and to create new variety is short time. My question is, how long you to create new variety with the new biotechnology, and how perform your variety property as quality, quantity that you create, and maybe to relate it to pattern of fit Indonesian people. Thank you.
Second.
Okay, Dr. Parawita, I think you have presented a really good comprehensive work there. I have one question. How long can you store those synthetic seeds?
Maybe in the back.
Okay, I have a question for Dr. Siti Nurbayoslan. And on this case, your research is get the optimum condition for the enzyme is 30 hours with 0.5%. And then, but I'm still wondering because the enzyme is lower than another treatment. It is right. I just wondering why the enzyme activity is
lower than another treatment. Okay.
I think 3 questions first. Okay, please. Or Trihandoyo.
Thank you very much, Pak Herman. I think this question may be for Professor Bambang beside you. Actually, in my research, I developed from the anti-culture. This process will reduce the 4-6 generation more fast than if we use the conventional breeding. And the problem if I compare with transgenic genetic engineering, maybe some target, random target. That's why you should choose the anther, good anther, and get the haploid and make the diploid or tetraploid and something by using chemical or electromagnetic treatment to get new varieties. And we also possible to increase the quality and quantity. I think that's my question. Thank you.
Dr. Parawita, maybe?
Okay, thank you for your question, Bu Norma. It's, I think, for, I can store it maybe until 3 months because it depends on plant growth. Because synthetic seed is encapsulated, we can get nutrition about macronutrient, micronutrient, maybe amino acid, and then plant growth. So to promote or to inhibit. So if no plant growth, maybe we can terminate, but we can give the inhibit plant growth retard like this retardant in my research. This is PEG, polyethylene glycol, maybe can terminate at 3 months. Yes, thank you.
Doctor.
Thank you, Riza, for the question. Okay, you asked why SO express T1 lipase lower than other strain. It is because we just use whatever Pichia pastoris have. In Pichia pastoris strain, we have a lot of Pichia pastoris strains, right? So this is a new strain that we don't know, and then nobody has used this as an expression host. So whatever optimization we are doing now is based on Pichia pastoris. That's why we are moving towards to understand what is this. So it may have different medium, different things later on. That's why maybe the problem is coming. That's why we have also metabolic profiling profile. We found that a lot of fatty acid, a lot of amino acid being released in the culture medium. Then maybe that one is also caused something inhibitor to the promoter to regulate the more T1 lipase to be expressed. Now we are hoping to understand more about this system because we know about whatever we know about Saccharomyces. Hopefully it can be implemented in this new system too. Hope this answers your question.
Okay, I think the time is off. This is a very good presentation. Okay, we can give the applause for all speakers. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
That was the end of invited speaker lecture for today. The next agenda is oral presentation. And we invite our moderator for this session is Ms. Intan Riana Liana. Ms. Intan, time is yours.
Okay, Assalamualaikum warahmatullahi wabarakatuh. Good morning. I will be served as your moderator for this sessions, and this session divides into presentation and discussions. And now I would like to invite the first presenter, Ms. Siti Nur Hasmah Ishak, to present her presentations. So, to Ms. Nur Hasmah Ishak, time is yours.
Okay, Assalamualaikum and good morning everyone. So my name is Siti Nur Hasmah Ishak and I'm a PhD student under the supervision of Professor Dr. Rajanul Saliha. So today I would like to present part of my PhD works, which entitled Structural Insight into Conformation and Stability of T1 lipase, Jebasinia zalihim mutant, and here again T1 lipase. So before I go deep into my research project, I will introduce about lipase. Okay, lipase is the enzyme belonging to triacylglycerol hydrolases, which involved in esterification, acidolysis, alcoholysis, and etc. And this lipase mechanism is it catalyzes the hydrolysis of triacylglyceride to free fatty acid and glycerol. And again, T1 lipase, the lipase that I work with is derived from Jebasinia zalihim, obtained from palm oil mill effluent in Malaysia. And this T1 lipase consists of 388 amino acids and composed by the catalytic triad serine 113, aspartic 317, and histidine 358. And this T1 lipase has been found to be optimum at pH 9 and also optimally active at 70 degrees Celsius. And this photo shows the three-dimensional structure of T1 lipase that has been elucidated in 2008 and has been deposited in Protein Data Bank as 2DSN. And lipase has been widely used in industrial application, such as detergent industry, which is remain as the biggest market for lipase industry, and as well as it also can be used to produce cheese flavor, to imitate the cheese flavor, and also can be used, can be utilized in the pharmaceutical and cosmetic industries. So I will just, okay. T1 lipase was sent to space under the program JAXA UPM Protein Crystal Growth Flight Program, and we sent to space for the crystallization process using capillary method, and the results are outstanding with the high crystal quality produced in both space and Earth, and space-grown crystal has been elucidated at high resolution, which is 1.1 angstrom and 1.3 angstrom for Earth-grown crystal lipase. And microgravity has equipped the excellent condition for the crystal growth with bigger crystal and better internal order, and it also leads to the higher intensity of the X-ray diffraction, and which also provide a better quality of electron density map. And you can see here the differences between space-grown crystal and Earth-grown crystal. And however, there is a limited discussion or limited study on the similarities and dissimilarities of the structure, architecture, and features in terms of hydrogen bond and ion pair interaction from the atomic model derived from this space and earth crystal structure. And usually the researchers only compare the crystal appearance is more beautiful in space, more bigger in space, better internal orbit in space, but they didn't study insight into the protein structure. So there's also the lack of manipulation on the structure information obtained for the production of new enzyme. So we don't know the characteristic of this enzyme. Hence, this study, my objective of my study is to re-engineer the new enzyme with improved characteristic with the information obtained from space crystal structure. And to achieve this objective, first I will compare the thermolipase structure obtained from birth condition, and then genetically redesign the enzyme based on the structure and identify the characteristic of this new mutated thermolipase. And the hypothesis of this study is the new enzyme obtained will be different in characteristics, and the addition of hydrogen bond and ion interaction might be increased to protein thermostability. And this is the methodology of my study, which the first part involved is in silico study of space and earth-grown structure using Yasara molecular modeling program. And from this study, I will identify the point of mutations and followed by the homology modeling using Yasara molecular program again, and using thermolipase template to build the structure of the mutant. And this mutant will be studied using the molecular dynamic simulation using GROMACS program, and also the essential dynamic study, which involves principal component analysis and free energy landscapes. And from this result, I will do the lab work where I mutate the original thermolipase using quick change lightning kit, and followed by the purification using affinity chromatography, and followed by the characterization where to determine the temperature, pH, and also the melting temperature using circular decreasing method. And the comparison of the space and earth structure, we can see that the result shows that space structure has a high hydrogen bond, however low number of ion interaction. However, after the simulation study, it shows that the interaction in space is stronger compared to the earth structure. From this figure, we can see that in earth structure, at the first part, before the simulation, it has the interaction. But after the simulation, the earth structure lost its interaction. This interaction is between the residue 226 and 230. But in space structure, this interaction is remain stable even though after 20 nanosecond.